Hello everyone, and welcome to this last part of the presentation. Now that we've established the knowledge of how cancer comes to be and how we should look at it given today's detection and treatment tools, let's talk about how nanotechnology comes to fill a gap left by the chemotherapy, radiotherapy, and the surgery that we discussed last time. We are going to talk about the size matters, which is basically defining nanotechnology before the red line, detecting cancer, supplementing chemo, supplementing radiotherapy, and supplementing surgery. We start, of course, by defining nanotechnology, which is simply put, technology of the small things. But then we, of course, have to get a sense of how small the nanoscale is. And let's say you have a 1 meter long piece of paper and you cut it into 10 equal pieces, right? So each one of those pieces has 1 decimeter. But if you were to cut the same piece of paper into a billion equal pieces, one would be 1 nanometer long. That's how small a nanometer is. A fully structure, to be a nanostructure, it has to have at least one of its dimensions measuring less than or equal to 100 nanometers. And this structure can be a nanorobot, it can be anything basically, but of course it differs from their normal size counterparts because the chemical and physical behaviors of those nanostructures take from such an incredibly small size. That's why we say the size matters. This second point before the red line reminds us of the other point in the second part of the presentation, which was after the red line, which was saying how cancer with today's diagnostic tools is detected when it's kind of too late because it was after the red line. But this is now saying how with nanotechnology we can detect cancer before the red line, giving us a chance of treating it perfectly. We do this by putting nanoparticles inside the body, programmed to look for cancer or any signs of cancer. And um, some of the ways they can do that is looking for carcinogens, right? We know a number of carcinogens, and we would be like, if you see this carcinogen, alert us because it will be a sign that there's a chance. They can also be programmed to watch over the product genes. Will be like, these are the kind of genes which, if mutated, give rise to cancer. So alert us if you see any change in the proton anchor genes. Or they can also look for cancer cells, given that the cancer cells have something in common, which is the presence of folate protein or the wider fenestrated blood vessels around them. If you see any of those, alert us. Of course, these are the things which do not require cancer to be after the red line to see, because even if it was one cancer cell in the body, the protein will be there and it will be knowing that the cancer is there and we treat it before it's too late. Or supplementing chemotherapy. One of the problems we've seen with chemotherapy in the last part is that chemotherapy kills healthy and cancer cells as long as they are rapidly proliferating. Nanotechnology brings a way of killing only cancer cells. We can use magnetic nanoparticles as drug carriers or as hypothermia agents. So in this case, nanoparticles have a magnetic core, and around the core there is a shell harboring a drug. These blue cycles represent drug, and they are inserted in the body. Due to the presence of a magnetic core, we can use an external magnetic field to drive those particles to a region of tumors. And after they are there, they can release their drug, killing uh, the cancer cells. With hyperthermia, it's almost the same, except that after we've driven them to the region of cancers and tumors, uh, there is no releasing of drugs. Instead, there is use of an alternating magnetic field, which makes uh, these particles to heat up and cause a localized temperature increase, which kills these tumors. But even if this tries to kill only cancer cells, there are still some few normal cells which are dead because it's driving them in the region of cancers and tumors. But in this region, like these cells on the interface can also die. If you want to be even more specific in killing only cancers, we can put cancer cell specific ligands on the surface of these particles, and they can only attach on the cancer cells exclusively and not any other cell. The nanoparticles can also play both roles, releasing drugs and inducing hypothermia to increase the success rate. Another problem with chemotherapy that we actually did not discuss in the last part is that cancer cells develop drug resistance towards chemotherapeutic agents. And again, and technology gives a way to counter drug resistance. Take an example here. There's a form of drug resistance in cancers which is caused by the overexpression of a DNA repair enzyme called MGMT. So once chemotherapeutic drugs destroys the DNA of a cancer cell, this MGMT repairs it and allows the cancer cell to survive. But the good news is there is another enzyme, BG, which destroys MGMT. So with that in mind, we can design a nanoparticle of this nature. There's a core harboring a drug, and there's also another shell, the second layer harboring the enzyme BG. So once inside a cell, this ruptures first and it releases BG. BG kills MGMT. After MGMT is dead, then the drug is released next, destroys the cancer cells, and there is no chance of surviving because MGMT, which would repair it, is destroyed. We talked also about supplementing radiotherapy, and the last time we talked about off-target radiation, which damage normal cells. Here, nanotechnology responds by regenerating the killed cells. So the nanoparticles deliver a retinoic acid to the subventricular zone, which is a region of the mammalian brain containing neurostem cells. These neurostem cells are the cells from which other cells of the brain grow and differentiate. So if some cells are killed, we can stimulate the neurostem cells to produce other cells. It is actually sounding like a fictitious Hollywood movie, but it has been done in mice. Nanoparticles deliver retinoic acid, which induced in vivo differentiation of neurostem cells in mice. So there's a chance, a hope that we can do the same in human brains. Oh, we talked also about supplementing surgery, and remember last time about the fluorescence induced in the brain, but also the autofluorescence property of the brain giving a false signal. Well, in technology here eliminates the chances of cutting healthy tissues, or because the iron oxide nanoparticles are used and these are surface functionalized, we add the ligands on the surface of those particles so they can only be uptaken, retained by the tumors. The tumors become clearly visible for 24 to 48 hours, which is enough time for a surgeon to cut off the tumors. We've also talked about the challenge of post-surgery monitoring of cancer relapse or recurrence. Here, nanotechnology gives an answer, which is nanoelectromechanical systems, NEMSs. Nanoelectromechanical systems can measure electronical and mechanical changes, but at a nanoscale. And why is this any good? It brings out mass resolution at a nanoscale. You remember cutting off the tumor, but also having to cut some part of the healthy tissue just to prevent the recurrence at the interface. Well, mass resolution at a nanoscale makes it that we can identify exactly where a tumor cell ends and where a normal cell begins, and we can eliminate the need of cutting the healthy part of the brain. So these nanoelectromechanical systems also have their deformation increasing with decrease in dimension, which means the smaller they get, the more they can measure accurately. And they can measure any small change as small as a breakage of hydrogen bond, which means during surgery, detect any small changes and give the surgeons a chance to know what's going on. And they also give quick responses like real-time monitoring. And even after surgery, the NEM cells, different from MLI, always watch and detect cancer recurrence, not needing an interval of 1, 2 months. And also different from implanted materials, they don't need to be removed later on because they were found to be biodegradable in the cerebrospinal fluid. I have a firm hope that at this point we have understood how cancer comes, how to decipher it and understand it in the context of the tools we have today, and then what would nanotechnology bring to the table to improve Thank you for following from the first part to this part.